How's it going, everybody? Welcome back to the channel. My name is Dakota Salazar. I'm a first-year medical student here at the University of Central Florida College of Medicine. And today I'm just going to be making a video on a couple old AP Bio questions that I gave to a class that I taught during a review session. And what we're going to be kind of looking at is the logic that you need to be using when you're approaching scientific questions, not only in college, but also on the MCAT. And so there's a certain level of reasoning that you need to get to that's way beyond just memorizing something. So we're going to work through these problems right now, and I'm going to show you some kind of things that I do and things that I would be looking for while I'm answering these questions, and show you how the knowledge kind of builds on itself. So with that being said, let's get started. So here's our first question. So the first question states, hemoglobin molecules contain a prosthetic group called heme, which contains an iron Fe2+ atom. This iron atom binds to oxygen, and the oxygen is carried to the muscles. Under acidic conditions, a positively charged amino acid interacts with a negatively charged amino acid, causing the release of oxygen. What type of bond is occurring between the amino acids in tertiary structure? Now, this question is extremely long and extremely lengthy, and it's really asking a simple question. But what test writers like to do, especially on the MCAT, as well as in class, is they'll give you these long vignettes, these long passages that contain a lot of information that you don't really need. So if I'm looking through this question, there's a few things that I see right off the bat that kind of catch my eye. So I'm going to go ahead and jump down to the question stem. What type of bond? So we're talking about a type of bond. It's occurring between the amino acids in tertiary structure. Tertiary structure, okay? And so it says, under acidic conditions, a positively charged amino acid interacts with a negatively charged amino acid. So let's reason through this question a little bit. So this question is kind of talking about the different levels of structure within a protein. And so if you remember protein structure, we have our primary structure, our secondary structure, and our tertiary structure. And primary structure is the sequence of amino acids. Sequence of amino acids. And those are held together by what? Peptide bonds. Okay? And peptide bonds are a form of covalent bond. Okay? Secondary structure is hydrogen bonding within the backbone. Okay? And hydrogen bonding is not a covalent bond. It is not a bond between hydrogens. All it is it's what we call an electrostatic attraction from a hydrogen atom to another more electronegative atom on another molecule. So the common example that they give is water hydrogen bonding to itself. Okay? And the reason water hydrogen bonds to itself is because the oxygen in water is more electronegative. So it's going to pull the electrons towards it, and we know electrons are what charged? They're negatively charged. So that creates a partial negative charge on the oxygen and a partial positive charge on the hydrogen. And these little weird symbols, those are delta, that just means partial. Okay? So partial negative charge on the oxygen, because it's more electronegative, it's pulling the electrons towards it, and the hydrogen is partially positive because it's losing those electrons. So what can happen is this is happening on two different waters. Okay? So this positive and negative can interact, and this positive and negative can interact. Okay? That is a hydrogen bond. And that's what's going on in secondary structure. Tertiary structure is interaction between R groups. Sorry for my terrible handwriting. So interaction between R groups. So every amino acid has an R group that comes off of its side chain, and those can interact. Those are things like positive and negative charges interacting, hydrophobic interactions, ionic bonds, covalent bonds in the case of disulfide bridges. We can also get hydrogen bonding and things like that. So looking back at this question, it says, under acidic conditions, a positively charged amino acid interacts with a negatively charged amino acid. Okay? When a positive and a negative interact, that's not hydrogen bonding. These are full positive charges. They're not delta plus or delta minus. They're full positive. So when we have a full positive and a full negative interacting, that is an ionic bond. So now that we've answered that question, let's go ahead and move on to the next one. So here's the next question. 2,4-dinitrotrophenol, 2,4-DNP, is an organic compound that disrupts the proton gradient across the mitochondrial membrane without disrupting electron flow. Which of the following is most likely to occur upon treatment with 2,4-DNP? Okay, so let's look at our answer choices. A, there will be a reduction in ATP synthase activity. B, there will be an increase in ATP synthase activity. C, oxygen won't get reduced to form water. D, A and C, E, B and C. Now these questions are kind of annoying because you need to know multiple aspects about the problem in order to get the question right. But they're also easy in the sense that we can eliminate answer choices pretty quickly. Okay, so let's take a look at the question. This compound, 2,4-DNP, disrupts the proton gradient. For those of you that don't remember, the proton gradient in the electron transport chain is when we transport H plus into the inner membrane space from the matrix, okay? And that's a form of active transport. We create this proton gradient where we get a lot of H plus built up in the inner membrane space, okay? So what that allows to happen is once that H plus builds up, it can come back down through the membrane through an enzyme called ATP synthase. I'm not going to write the whole name out, but that's ATP synthase. And when the protons flow back down through ATP synthase, it creates ATP. Okay? So the establishment of that proton gradient is directly linked to the synthesis of ATP. So if we disrupt that proton gradient, and what 2,4-DNP actually does is it actually kind of pokes holes into the membrane so that these H pluses can come back through it without going through ATP synthase. Okay? But if we disrupt that proton gradient, we disrupt ATP synthesis. So there will be a reduction in ATP synthesis activity, or ATP synthase. Okay? Therefore, B is wrong. We will not see an increase in ATP synthase, we will see a decrease. Therefore, E is wrong because that contains answer B. So now we're between three choices, and all we really need to look at is whether C is correct or not. Oxygen won't get reduced to form water. Well, when does oxygen get reduced to form water? What does it mean to reduce? To reduce something means to gain electrons. So, the whole point of oxygen being the final electron acceptor, I'm sure you guys have heard that before. Oxygen being the final electron acceptor just means that when the electrons get dumped off of our electron carriers, those being NADH and FADH2, they can flow through the electron transport chain, land on oxygen, and reduce it to form water. Okay, oxygen gaining those electrons reduces to form water. This compound does not disrupt without disrupting. It does not disrupt electron flow. Therefore, we will still see our water or our oxygen getting reduced to form water. So, answer C is not correct, and that leaves us with A. Let's move on to the next question. Question 3. The enzyme pyruvate dehydrogenase consists of an amino acid with an acidic R group. However, when this amino acid is switched with another one that has a similar R group, the overall structure of the protein remains intact. Which answer best explains this? This is another question on protein structure. And so what essentially this question is saying is we've taken an acidic amino acid in the structure of this enzyme, and we've replaced it with another acidic amino acid. Okay? So, for example, we took glutamate and exchanged it for aspartate. Those are two acidic amino acids. That's a type of mutation or change that we can have. So which levels of protein structure will be affected? So A says the switch of the amino acids had no effect on the secondary structure of this enzyme. Well, what's secondary structure? If we go back 2 slides, Secondary structure is hydrogen bonding within the backbone. Hydrogen bonding is not dependent on R group. So we could have changed the amino acid for a completely different amino acid, and we would still have no effect on secondary structure because it's just hydrogen bonding between the backbone. And if you recall, every amino acid has the same exact backbone. So A is true. B, the switch of the amino acids had no effect on primary structure. Well, what's primary structure? That's the sequence of amino acids. Okay? Did we change the amino acids? Yes, we did. So that did affect primary structure. So B is out. Therefore, E is out. C, the switch of the amino acids had no effect on tertiary structure. Tertiary structure is the interaction of the R groups with other R groups in the enzyme or protein. We switched an acidic R group with another acidic R group. There is no effect on the tertiary structure of this enzyme because those R groups are able to interact very similarly with the groups that they were around previously. Does that make sense? If I would have changed this R group for something else that wasn't positive or negatively charged or acidic, then it would have some effect. But this is okay. So therefore, D is the correct answer. So moving on, I'm actually going to skip number 4 and jump to 5. So 5 says, cyanide is a potent chemical that binds tightly to human hemoglobin, displacing oxygen. Cyanide is targeted to the brain tissue where it binds to the enzyme cytochrome oxidase and shuts down the electron transport chain. Which of the following would you expect to see in a patient with cyanide poisoning? Okay, so this question I'm really trying to make a point, but let's reason through it. So the electron transport chain is responsible for what? Well, we already said it generates ATP. Okay, generates ATP. Well, what happens when we shut down that electron transport chain? If we shut down the electron transport chain, we have to go through anaerobic respiration. Okay? And anaerobic respiration essentially just means we're taking that pyruvate that we made in glycolysis and we're converting it to lactic acid or lactate. Okay? So, if we don't have any oxygen at that electron transport chain, we can't create ATP. Therefore, it makes no sense for us to keep shuttling this pyruvate to form acetyl-CoA into the TCA cycle, creating all these electron carriers when they're just not going to be able to do their job. So what happens is we shut down TCA and we shut down the electron transport chain. And what we get is this pyruvate being converted into lactic acid. So we would see increased brain lactic acid concentration. A decreased brain ATP levels. Yes, because we're not able to do the electron transport chain. If we're not able to do the electron transport chain, we're not generating a lot of ATP. If you remember, the electron transport chain makes about 32 to 36 ATP. That's a big chunk of ATP. If we're not doing that, there are going to be way low levels of ATP. So B is correct. Okay? When these two answers are correct, I'm immediately looking for an answer choice that either says A and B or all the above. As soon as you know that two are correct, go to all the above, and it doesn't matter. Okay? And the reason I threw D in here is because oftentimes they'll throw in an answer choice that really doesn't make sense and it's not familiar to you, but it's correct. But they just want to test whether or not you'll trust your gut and go with the answer choice that you've stuck with to begin with, because oftentimes you're going to be right. Okay? So as soon as you see that more than one of the answer choices are correct, you need to look for all the above. And so moving on to our last question. Question 6. During periods of restrictive carbohydrate intake, our liver starts to convert fatty acids into ketone bodies. These ketone bodies are picked up by the surrounding tissue and converted into acetyl-CoA. If a 16-carbon fatty acid was broken down into 8 ketone bodies, how much NADH and FADH2 would be produced respectively? Okay, so let's break this question down. Our question stem said that ketone bodies are converted are converted into acetyl-CoA. So if we have 8 ketone bodies, how many acetyl-CoAs will we have? We're going to have 8 acetyl-CoAs. Okay. Acetyl-CoA can enter in cellular respiration at what step? It can enter in at the TCA cycle, or Krebs cycle, whatever you want to call it. Okay? When it enters the TCA cycle, per cycle, remember, it's a cycle that regenerates oxaloacetate. I don't know how to spell whatever. It regenerates oxaloacetate. Acetyl-CoA can enter in, and it goes through this whole cycle, creating 3 NADH and 1 FADH2 per cycle. Okay? 3 NADH and 1 FADH2 per cycle. If we have 8 acetyl-CoAs, all we need to do to get total numbers is multiply these by 8. So we'll get 24 NADH and 8 FADH2. That's answer C. I hope these questions were very informative for you guys, and you see how you should be working through these problems and thinking through them. If you have any questions, please feel free to reach out to me. But with that being said, do not feel discouraged if these were a little difficult for you guys. These are meant to be difficult. They're probably going to be harder than what you're going to see in class. And this is more kind of biochemistry level knowledge. But you're still going to learn a lot of this in Biology 5A and those bio classes that you're going to take during your first year.